students, in this video we will start cell cycle. Okay, we will discuss about cell cycle. Start. So what is cell cycle? Cell cycle is the order of series of events. That means it is the sequence of events by which a cell duplicates its genome and eventually divides into daughter cells. Okay, again I am repeating the informations. Cell cycle is the sequence of events by which a cell duplicates its genome and eventually divides into daughter cells. Okay, so in a cell cycle there is two phases. One is interphase, another is M phase. This is the graphical pie diagram of cell cycle. Okay, here is the interphase. It has three parts: G1 phase, S phase, G2 phase. These are the subphase of interphase, and this is the M phase. In the M phase, the cell actually divides, and this M phase is consists of two parts: karyokinesis and cytokinesis. And this karyokinesis has two parts, so four parts: prophase, metaphase, anaphase, and telophase. And after that, the cytoplasmic divisions of cell occur and two new daughter cells are produced. So this is all about the shortcut idea, gist of cell cycle. In simple word, you will say what is cell cycle. साइकिल का मतलब ये होता है जहां से शुरू होना फिर वहीं पॉइंट पे पहुंचना. Where we start, again reach at that point. This is called cycle, and here the events that are occurring in a cell is such a cyclic event. After cell division, think new daughter cells are produced, and now in that daughter cells the activities will occur. Now this cell will again prepare itself for dividing. Okay, so you can say cell cycle is the time gap. It is the time, it is the period in between a new daughter cells produced and again it prepares itself to divide to form a new daughter cells. Okay, so just think what happens in interphase. What happens in a cell division? From a single cell, two cells are produced. So एक से दो होना चाहिए. That means the cell is converted from 1 to 2. So inside the cells, the materials, the cell organelles, the protein, the DNA content, which are present and needed to be doubled. Okay. And after that the cell can be divided. The cell should stock the materials that is essential for the formation of two new cells. So this is the interphase where the cell prepares its materials for the formation of two new cells from it. In G1 phase, the cell, before discussing about G1, S, G2, I am mentioning simple tricks so that it will be easier to understand. In S phase, the DNA replications occur. So S phase is known as synthesis phase or synthesis phase. So here DNA replications occur. If in S phase the DNA replication will occur, definitely in G1 phase the synthesis of those materials are essential occur which are essential for DNA replications. The enzyme which are used in DNA replication should be gathered in G1 phase. So in G1 phase, a number of enzymes, number of proteins are synthesized which are highly essential for DNA replications, and some other things also happens in G1 phase. And G1 phase is the most longest phase, subphase in the interphase. Okay. After that the S phase, in synthetic phase, the DNA replications will occur. Okay. In DNA replication phase of S phase, DNA replication occur, but those nucleic acids remain attached to the template DNA. Although there is DNA replication occur, but there is no increment in the chromosome number. The separations of chromosomes or chromatic or nucleic acid will occur in the nuclear divisions or karyokinesis phase. And also the histone protein synthesis starts at the beginning of the S phase. After that the G2 phase occurs. In the 3 interphase, G2 phase is quite subtle than other, but it is highly important because in G2 phase there will be checking of any defect or any error that occurred in DNA during the S phase of DNA replications. So if there is any error, then the DNA repairing will occur in G2 phase. Okay. After checking all these things, the the cell will proceed, the cell cycle proceeds toward M phase. In the M phase, there will be first karyokinesis, then cytokinesis. In karyokinesis, there will be start like prophase, metaphase, anaphase, telophase, and after the completion of karyokinesis and 2 new nuclear formations, cytokinesis will occur. Okay, this is all about cell cycle in gist. Actually, you are reading this cell cycle from the very beginning of 9th, 10th in school or in plus 2 also. The actual concept behind this cell cycle is the cell cycle controlling system. How the cell cycle is controlled. What are the points, what are the proteins, what are the mechanism that controls this cell cycle. For example, if in G1 phase sufficient amount of proteins and equipments are not found, that is essential for S phase DNA replication, the cell cycle will not proceed towards S phase. There is some checkpoint, there is some checker, there is some protein, there is some mechanism which will not allow the proceeding of cell cycle from G1 to S phase. Okay, we need to clarify that things. This is the gist in higher classes. And even in S phase, after DNA replication, if there is any problem in DNA replication or any type of error in DNA replication, that will be checked in G2 phase. And that G2 phase has various kinds of checkpoint and mechanism. For example, ATM mechanism, ATR mechanism, checkpoints mechanism, those will detect whether there is a problem in the DNA that has been replicated during the DNA replications of S phase. So they will allow or not towards the M phase. So the important thing is cell cycle control. Okay, which we will discuss in the next video. Today we just discuss what is cell cycle, which is the sequence of events by which the cell duplicates its genome and eventually divides into two data cells. And in another thing I have to mention here, in some cases the cell cycle after completing the M phase, when new data cells are produced, some cells enter into a special phase called as G0 phase, where there is pause in the progressing of cell cycles occur and the cell remain in that state until it start resuming of further cell cycle process or proliferation process. Okay, that means in the experimental found that in a typical rapidly mammalian proliferating cell human culture, the G1 phase consisting of 11 hour, S phase 8 hour, G2 phase 4 hour and M phase 1 hour. This is just an idea to clarify you that interphase has maximum time 23 hour and M has 1 hour. Cell to cell, species to species, the cell cycle duration varies. Okay, it is not fixed in every cells. Even if the interphase duration in human cells in any human cells is not same. Okay, you all now clear the concept. Okay, so what is G0 phase? Again, G0 phase is the phase that found in some cells which withdraw themselves from further proceeding the cell cycle. Actually, one curious thing is that in maximum human cells are in G0 stage. Skeletal muscle cells and the neural cells of human body are in G0 phase. Okay. For further video update about the cell cycle controls and different checkpoints, please like, share and subscribe my channels in order to get the video in a sequence manner and update.